Hi guys, welcome to another instalment of Biology with Miss Cherry. As you can see, we're changing pace a little bit today, so we're no longer talking about gene mutations, and instead we're going to take a look at different cell types. So take a look at the picture. What does it show? And what is the difference between totipotent, pluripotent and multipotent stem cells? See if you can get any more information from this diagram. Maybe you fancy doing your own research, but pause for a few minutes and then carry on once you've had a think about those questions. So as I've said, or as you should have seen from the starter, today we're taking a look at stem cells. So we're going to think about what our totipotent stem cells are and how stem cells lose their totipotency to become slightly more specialised, so into pluripotent, multipotent and unipotent stem cells. We're going to have a look at their uses and maybe how we can do some kind of super cool biochemistry to take a specialised cell and revert it back to a more potent stem cell type. So let's get into it. In terms of our cells, all of our body cells have got all of the same genetic information. So each of our cells has got exactly the same 46 chromosomes in 23 pairs in them. And this is because all of our cells come from the same mitotic divisions of a single fertilised egg, which we're now going to call a zygote. Okay, so when a sperm and an egg come together, that fertilised egg is then the thing that develops into the fetus and then the baby. And I want you to think about this question. If all cells are genetically the same, how are they different? So how are the cells that make up my hands different from the cells that make up my liver, that are different from the cells that make up my heart, that are different from the cells that make up my white blood cells? How are they different? Because technically they should all be the same. So pause, maybe do some research, look at the diagram, it should give you a little bit of help. And you can see in the picture that we have to switch some genes on and off. And this is a process called epigenetic changes, so gene regulation and epigenetics. And this is a topic that we're going to take a look at in a few lessons' time. But the genes that are switched on and off in each cell type is different. So you can see in this diagram that from a single cell, if we're going to develop a neuron cell line, then we're going to start expressing neuron-specific genes, whereas if we're producing skin cells, epithelial cells, then we're going to be able to start producing epithelial-specific genes. There's no point, and it's a waste of cellular energy, if that epithelial cell starts producing neuron-specific genes and vice versa. And when we think about our cells, although there are differences, we also need to think about what is important to happen in all cells. Which genes are important to be on or conserved or expressed in all of our cells. Okay, so spend some time thinking, making some notes, what kind of processes need to happen in every cell. So pause, and then when you're ready, you can play again. So all of our cells are going to undergo key metabolic processes. Okay, we know that in all of our cells, we're going to need to undergo things like respiration. So we're going to need to be able to produce key enzymes for respiration. So we're going to need proteins for things like enzymes, but also the proteins for the electron transport chain, the proteins for the movement between the cytoplasm and inside of the mitochondrial matrix, the things that move the products of glycolysis into the cell. Okay, but we're also going to need to produce enzymes that are involved in transcription and translation. And next lesson, we're going to have a look at how transcription and translation can be regulated. Okay, so remember, in all cells, we need genes controlling key metabolic processes, your respiration, your photosynthesis if you're a photosynthetic organism, and those involved in transcription, translation, and general cellular functions like protein synthesis. We're also going to need to be aware of the fact that some genes are not important in certain cell types. Okay, so if I think about my beta cell from the pancreas, maybe if we're talking about diabetes, for example, that's important that the beta cells are able to produce insulin. Those genes for insulin production are switched on. My eye has got exactly the same genes in it, but I'm not going to need to produce insulin out of my eye. That's not the function of that specific type of tissue. Okay, again, it's important to be aware of what is useful in one cell type might not be useful in another. Now, in terms of genes being switched on and off, in some tissues, so if we go back to the eye and insulin producing genes, some genes are switched off or on permanently. Okay, so if a cell's sole function is there to produce maybe some digestive enzymes, such as maybe one of the cells lining the stomach, they're going to have the genes for acid production on all the time. It's going to constantly be there. It's part of its function. Some cells, though, depending on the needs of the cells at specific times, will switch genes on or off. It saves energy, okay? So it stops wasteful production of protein. Because remember, if you think about it like baking, all of these proteins that are going to be made need ingredients. So the body needs to do these processes, and it takes time, it takes energy. And if we're in an energy conserving state, maybe we don't have a lot of food around, because other organisms will do this as well, we don't want to waste that energy. Okay. And don't forget that DNA helps to make the amino acids, which come together to make proteins, and that gives us the phenotype. So when we're talking about these switching genes on and off, we're really affecting all of the downstream processes as well, that amino acid production, proteins, and the phenotypic expression. From here on in, we're going to take a look at these stem cells. So we mentioned in the starter about totipotency, pluripotency, and multipotency, and hopefully you might have heard about those at GCSE, but I am also conscious of the fact that GCSE for you guys at this point was nearly, or it was probably 2 years ago. Okay, so we'll start from the beginning. We'll go from a very, very basic overview. When a sperm and an egg come together, that zygote, that fertilised egg, is formed. And throughout the pregnancy, through the duration of the developmental stages, that egg ends up a full human. Okay? And a zygote, therefore, has the ability to turn into any cell type. And we say that the zygote is made of totipotent stem cells. Okay? Totipotency, or total potency, they have the potential, okay, to turn into anything. If we split that word up, total potential, to become anything, any cell type. Okay, and again, that egg and the sperm come together, and they're totipotent, but also the original few cells that we produce to make, like the blastocyst, for example, in early development, are also going to be totipotent. And totipotency occurs in animals, so us as we're talking about this zygote and developmental stage, but also in plants as well. And you need to be aware of that, because exam questions will often try to trip you up and ask about totipotency in plants, when really lots of people do just forget about it. So we can see here an example of cell differentiation from totipotent to pluripotent to multipotent stem cells in animals at the top and in plants at the bottom. And you can see in both situations that our totipotent cells are only present at the very beginning of both of these cell differentiation diagrams. Okay, immediately after blastocyst formation in humans and original cell differentiation in plants, the stem cells start to lose some of their potency, so they become pluripotent, and we'll look at what those means, and then they become multipotent or unipotent. And again, if you haven't heard about those before, we will take a look at those in a second. Okay, so totipotency is only at the very beginning of development. And as we said, in that diagram, the totipotency occurs only at the very beginning of the developmental stage. And after that, the cells become specialised, so they start to lose that potency. Now at A level, we're going to change our definition of a specialisation. At GCSE, it was a cell able to do a specific job or suited to do a specific job. But this time you have to say that specialisation is where a cell type is adapted to a specific function, and the key A-level difference is because only certain genes are expressed. Okay, now, I've told you already that we're going to have a look at factors affecting gene expression in the next lesson, and it's controlled by mechanisms that limit or prevent transcription and translation. So switching genes on and switching genes off by changing the structure of the DNA. Now have a go at doing some of your own research about how cells specialise. Generally we say that specialisation of the stem cells is mostly irreversible. So if I've got a pluripotent stem cell and it ends up going to make, I don't know, some digestive cells, for example, you can't generally say, all right, okay, I'm done making digestive cells because I've made a whole organ. I just want to go back and become a little bit less specialised again, and I'll maybe go and make some liver cells. It's not how it works. So have a go at researching these 4 questions. What is meant by stem cells? What's special about them? Where are the stem cells found in mammals, not just animals? And what's different about each stem cell? So that's your toti, pluri, multi, and unipotent stem cells. Do some research. We're going to take a look at each of those in a sec, but pause now and then play when you finish those. Here's a little task for you guys to do as well. So you can use your books if you've got them and use the internet. And on the next slide that I'm going to change to in a second, there are lots of statements. They should be in a grid formation. I'll show you what I mean in a second. But can you please sort them out into piles of properties of totipotent, pluripotent, multipotent and unipotent stem cells? Okay, give yourself about 5 minutes. Hopefully it should be nice and simple if you've done some good research for the previous question, but you might need to spend a little bit more time just writing them out and comparing them in a bit more detail. So here you can see the statements that I need you to organise, to sort. As you can see, we've got the headings for each of your columns in the highlighted red, and the highlighted green shows the bullet points, the notes, the information that you need to know about each of these types of tissue. We are going to take a look at them in a bit more detail, each individually in a second. So if you're not too sure about any of these, you're welcome to just carry on watching the video. Otherwise, pause now and try and organise each of these into their columns. So the first type of stem cell that we're going to look at is totipotent. These are the ones that are the most unspecialised, and these are the type of cells that can develop into any cell type. So we say that they're found in the developing zygote, so after that fusion of the sperm and the egg cell, and the morula, which, as you can see in this diagram, is the early developmental stage of the embryo before it starts to become slightly more specialised in the blastocyst. And during development, so as you can see even in this little diagram, the stem cells become less potent, they have less potential to develop into other cell types. So even moving from the morula to the blastocyst's inner cell mass, so not the blastocyst itself, those stem cells are now pluripotent. So they've lost some potential to become all cell types. Pluripotent stem cells are found in the early embryo and as a fetus develops, and these can become almost any type of cell. So they can't become a full human, but they can form all of the tissue. So if a pluripotent stem cell breaks off, it's probably just going to die. Whereas if a totipotent stem cell part of that morula starts to break away, that's generally how we become twins. Okay, so these pluripotent stem cells can become almost any type of cell. And these are found in embryonic stem cells and fetal stem cells. So those are your 2 examples of the cell types that you need to be able to mention. Multipotent stem cells are the ones that are found in adults, but these are limited. So you'll see how we've gone from toti, so totally potent, plurally potent, maybe there's lots of different cell types that they can become. Multi is slightly less, so we're limiting the number of cell types that we can become. In this example, we've got things like bone marrow stem cells, okay, and these can become any type of blood cell. Those bone marrow cells are called hematopoietic stem cells, okay, and they are found in the umbilical cord. So when we say adults, we're also talking about like fully grown babies, and also in the bone marrow of the long bones, so things like the femur, things like the pelvis. And finally, we've got those unipotent stem cells, and this is when a cell type can only become a single type of cell. So you can see here in the diagram that our multipotent hematopoietic stem cell is able to become red blood cells. Then those red blood cells are able to divide to become more red blood cells. It's important to recognise that not all cell types are unipotent. It's normally for important cells, things like neurons and blood cells that have got key biological roles. So now we're here, we've had a think about the different types of stem cells. Could you now stop for a second, pause, and suggest a reason why skin cells retain an ability to divide by being unipotent when the cells of some other organs don't? So think about where the skin is, think about why those skin cells might need to continue dividing. Use your common sense, it's not as tricky as you think it is. So pause now, spend a couple of minutes, and then we'll go through the answers. So hopefully you should have been able to recognise, because you are made some part of skin, that skin is on the outside of your body. It's the external surface, and therefore it's likely to get damaged. And because it gets damaged, it's going to need to be replaced more frequently. Okay, if you lost all of your skin cells and didn't replace them, you'd very quickly be like a big jelly blood bag. Conversely, those internal organs, things like your heart, your liver, your kidneys, are less prone to damage. They're not likely to be as subject to the external environment as the skin is. And also, they've got other protective mechanisms, things like fatty layers around the kidneys, for example, or sort of other organs and bone structures protecting them, things like the sternum for the heart and the ribs. Yeah, so they're not going to need to replace as regularly. That isn't to say that they can't. Things like the liver can regenerate itself, but they're less likely to have to be able to do it. Now, there's a new type of stem cells which is quite exciting, and these are called induced pluripotent stem cells. You can abbreviate it to little iPS, and this is a bit of a complicated one because we're effectively reversing unipotent cells to turn them back to pluripotent cells. And we're effectively working to reprogram, to change the genetics of those unipotent cells in order to help them turn back to those pluripotent stem cells, which we can then use to increase the proliferation of any cell type that we need. And we're thinking about this therapeutically. It's going to be really useful if you've got a condition that is degenerative. Maybe a certain type of cell has been broken down over time, or maybe it's not working as well. Now, this is a really, really complicated process. You can see in this diagram that we have to apply a lot of chemicals, things that are going to help gene induction, so increasing the formation of gene products, and also transcription factors, which we'll look at next lesson, as I've said, to help genes turn on and turn off. And all of these chemicals, so SOX2, C-mice, all of that stuff is going to be able to help reprogram those cells and turn them into more cell types than they would have otherwise been able to. Now, these iPS cells are very, very similar to the embryonic stem cells, the ones that we get from the umbilical cord, for example, but they're not exact duplicates because, so let's think of the example, I don't know, we'll use frogs. Okay, if I've got some frog spawn, that can turn into a tadpole, and then the tadpole will turn into a frog. Okay, that's how the development happens. So embryonic stem cells are the frog spawn, and then they're going to turn into the multipotent stem cells, which are maybe the tadpole, and then the tadpole turns into the frog, which is a unipotent stem cell. However, if I take a frog and I try to apply chemicals to reverse it, even thinking logically, that's probably not going to give me the same kind of frog spawn. It's probably going to be a little bit funky somehow. Okay, but the interesting thing about these iPS cells is that despite being specialised as those unipotent stem cells before they reversed, if we turn them into an iPS cell, they're able to regenerate. So they can actually undergo mitosis. And now normally, you should have seen in the mitosis lesson in module 2, when a cell specialises, it can't undergo mitosis, it leaves the cell cycle. So if we've got these regenerable cells, then there's the potential that if they can continue to divide, we can get a whole collection, a whole sample of iPS cells, which means that we don't have the ethical concerns associated with using embryonic stem cells, because these iPS and embryonic stem cells should have the same kind of properties. Now, if you're in my class and we're doing this as a distance learning lesson, could you please have a go at these tasks? If you are not in my class and you're just watching this video, then these are from the Oxford book, and it's the Year 2 specific one. If you haven't got the Year 2 specific one, then it will be sort of around page 500 in the 2-year books. Okay, so pause now, have a go at these tasks. Remember, we've got to think about plants as well, and then we'll carry on. So for the final bits of the lesson, as per usual, we're going to have a look at some exam questions. So plant physiologists attempted to produce papaya plants using tissue culture. They investigated the effects of different concentrations of 2 plant growth factors, thinking about topic 6 again, on small pieces of the stem tip from a papaya plant, and the results are shown in the table. So we've got concentration of auxins down the left-hand side and different concentrations of cytokinins going sideways. Okay, so this is the background of the question. We're trying to grow different parts of plants using tissue culture. A callus is a mass of undifferentiated plant cells, whereas plantlets are small plants. Explain the evidence from the table that the cells from the stem tip are totipotent. Explain the evidence from the table that cells from the stem tip are totipotent. Use your knowledge. Think back to what totipotent means. Have a go at this question. It's 3 minutes, so pause and then play again when you're ready for the next one. Now this next question is a 2-mark calculation. Calculate the ratio of cytokinins to auxins. Be careful of the way they want that round. Cytokinins to auxins that you would recommend to grow papaya plants by this method. Okay, so think about what the difference between a callus and a plantlet is. Think about which ratio would be the best one to grow papaya plants. 2 marks, 3 minutes. Play when you're ready. Papaya plants reproduce sexually by means of seeds. Papaya plants grown from seeds are very variable in their yield. Explain why. So think about what you know. It might not be related to the table. So you need to maybe use your knowledge. Why might papaya plants that are reproducing sexually have variable yields in the plants that they've produced? 3 minutes, pause and then play again. Explain the advantage of growing papaya plants from tissue culture rather than from seeds. So you're going to need to think back to your GCSE knowledge about tissue culture. Hopefully you will have learnt about tissue culture in the independent work section of the lesson when I had those 3 tasks on the board. If not, maybe go back, find a GCSE science video on the YouTube, and then you should be able to answer this question really. It's not too tricky. Now here we're on a different concept, so we've moved away from the plants now. SCID is a severe inherited disease. People who are affected have no immunity. Doctors carried out a trial using gene therapy to treat children with SCID. Doctors who carried out the trial obtained stem cells from each child's umbilical cord. Give 2 characteristics of stem cells. 2 marks. I'll give you 2 minutes for this, so pause for a couple of minutes, have a go at these questions, and then play again in a second. The doctors mixed the stem cells with viruses. The viruses had been genetically modified to contain alleles of a gene producing full immunity. The doctors then injected this mixture into the child's bone marrow. The viruses that doctors used had RNA as their genetic material. When these viruses infect cells, they pass their RNA and 2 viral enzymes into the host cells. One of the viral enzymes makes a DNA copy of the virus RNA. Name this enzyme. So you might want to think back to HIV for this one. Just a hint. Pause. Play again in a minute. The other viral enzyme is called integrase. Integrase inserts the DNA copy anywhere in the DNA of the host cell. It may even insert the DNA copy in one of the host cell's genes. The insertion of the DNA copy into one of the host cell genes may cause the cell to make a non-functional protein. Explain how. So think back to last lesson. Think back to what we've done. Okay, you've got this, you can do it. You just need to think a little bit wider than outside of this lesson. And the final question, some of the children in the trial developed cancer. How might the insertion of the DNA have caused cancer? So this is a little bit of a tricky one because we haven't done a cancer lesson yet. You will have done cancer in topic 2, module 2, but there's another cancer lesson coming up in a few lessons' time. So you might get this right, you might not get this right. Don't worry about this one just yet. Maybe come back to it after you've done the cancer lesson. Oh, no, this is the last question. 5 out of the 20 children in the trial developed cancer. Although this cancer was treated successfully, the doctors decided to stop the trial in its early stages. They then reviewed the situation and decided to continue. Do you agree with their decision to continue, and explain your answer? So do you agree with the fact that although some of the children developed cancer, the cancer was then treated and the doctors decided to continue the research? Do you think that's a good decision? Why or why not? Okay, now we're going to have a look at the answers. I'm not going to talk through these ones. I'm just going to put the answers on the board. You can pause for now. If you've got any questions about the mark scheme, please let me know, and I'll try to answer them as best I can. But these are for the first few questions about plantlets, or the plants and the papayas. And here we've got the answers for the beginning of the SCID question. So about stem cells and the enzymes and how we can get different proteins. And then the final marks are about the cancer itself. So how does the cancer occur? How could the cancer occur if you're inserting some DNA into a gene? And then finally about should they continue, yes or no? And there's a couple of reasons why. So remember, for these mark schemes, each separate mark is shown by a semicolon. And if a word is underlined, you need to have all of the marks or all of the underlined bit for the mark to count. Okay, I hope you found this useful. Let me know if there's anything else you need me to cover regarding this, and I'll see you next time. Bye!